Hello everyone, welcome back to my channel Anytime Medicine, and today we are going to talk about is DNA replication. So what is replication? Replication just means making a copy. So DNA replication means making a copy of DNA. Eukaryotic replication is more complex than prokaryotic replications. Both uses many enzymes analogs that we are going to see in further slides. In both, DNA replication is semi-conservative, involves continuous and discontinuous synthesis. In discontinuous synthesis, we can see Okazaki fragments that we are going to talk about in further slides. Remember that replication occurs in 5' to 3' direction. So first thing we are going to talk about is origin of replication. Origin of replication is where particular sequence in genome from where DNA replication can begin. It may have single strand or may have multiple strand. If the cell is prokaryotic, it will have single strand, or if the cell is eukaryotic, it will have multiple strand. We can see the Tata box where you can find 80 rich sequences, which is adenine and thymine rich sequences, from where replication of gene can begin. So next we are going to talk about replication fork. Replication fork is Y-shaped region along with DNA template where leading and lagging strands are synthesized. You can see lagging strand here and you can see leading strand, it is here. So next thing is helicase. Helicase unwind DNA fragment and replication fork. You can see in the picture that helicase is unwinding the DNA in order to replicate the strand of DNA very easily. If we have deficient of this enzyme, we will get the Bloom syndrome, and that is the mutation of BLM gene. So next thing we are going to see is single-stranded binding proteins. Those will prevent strand from reannealing. You can see the structures of single-stranded binding proteins. They are attached to DNA segment and that will prevent DNA from binding it again. So next component is DNA topoisomerase or DNA gyrase that will create a single or double-stranded break in the helix to add or remove supercoil. You can see in a picture that DNA gyrase is further ahead from helicase and breaking the super bonds that are so strong for helicase to break, and that is making helicase work so easy. Further in DNA topoisomerase, there are some inhibitors that can stop topoisomerase. In eukaryotes, irinotecan and topocan inhibits top1 and adiposide and teniposide inhibit top2. In prokaryotes, fluoroquinolones inhibit top2, which is DNA gyrase that we have seen in previous slide, and top4. Top stands for topoisomerase. So next enzyme is primase, which makes RNA primer on which DNA polymerase 3 can initiate replication. You can see the location of primase, which is allowing DNA polymerase 3 to initiate replication. Now we have already mentioned DNA polymerase 3 in previous slide, so let's now talk about it. Only prokaryotes have its elongates leading strand by adding deoxynucleotide to the 3' end and elongate lagging strand until it reaches the primer or preceding fragment. Here you can see the preceding fragment of this Okazaki fragment. There are certain things for DNA polymerase 3 that you really have to keep in mind. That DNA polymerase 3 has 5' and 3' end synthesis, and that proofreads with 3' end to 5' end exonuclease. Drugs blocking DNA replication often have modified 3' end hydroxyl, that thereby preventing addition of the next nucleotide, because that will have hydroxyl group in the terminal nucleotide that will prevent the replication, and that is called chain termination. So now we are going to see is DNA polymerase 1, which only prokaryotes have. It will degrade the RNA primer and replace it with DNA. It will have same function as DNA polymerase 3. It will also excise RNA primer with 5' end to 3' end exonuclease. We have seen 3' end to 5' end exonuclease in DNA polymerase 3, which proofreads the DNA. You can see the structure of DNA polymerase 1 that is located here. You can see this red area, which is RNA primer. It is replaced by this blue area. With the help of RNA polymerase 1, this blue area is DNA. Now we are going to see is DNA ligase, which catalyzes the formation of phosphodiester bond within a strand of double-stranded DNA that joins the Okazaki fragment. You can see the gap between these two Okazaki fragments that is attached by DNA ligase here. So now we are going to see is telomerase, which only eukaryotes have it. In cell division, normal cells capable of only 60 to 70 divisions only. Then thereafter they become senescent. This is caused by shortening of telomeres. What telomerase can do is that this will avoid the shortening of telomeres and therefore the avoid of genetic loss, and this will dysregulated in some cancer and allows unlimited replication. This dysregulation more often will be upregulation. So telomerase can do is making the cell immortal, and that will allows the replication go further and further. So that concludes our video on DNA replication. Thank you everyone for watching. Do subscribe my channel, share it to your friends, and see you until my next.